Well, I'm not sure it is the best candidate. Okay, so one of the important ways that organisms interact is through chemical signals in terms of identifying potential mates, identifying your species, perhaps saying something about your fitness. And when I was studying sexual behaviour in Drosophila, because it's an interesting character, it's obviously got some fundamental evolutionary significance, so I started studying this in the second half of the 70s, and we assumed, it was known, it had been known for since Morgan started working with Drosophila, there was clearly something going on, because you could say, you knew that if you put a pair of flies to mate in a tube in which flies had already mated, so if there's a dirty tube, who knows what that dirt consisted of, but it wasn't a clean tube, mating took place more rapidly. So we knew there were chemical signals, but nobody had any idea what they were. And there have been various attempts to try and study them, but it was very difficult. So I went to France to try and understand various patterns of mating between species on the basis of their pheromones, or some aspect of their pheromones, which were studied, have been discovered in the early 1980s, had initially been found in Musca domestica and the housefly, and then people in France started looking at similar compounds which are on the surface of the fly in Drosophila. The great advantage of Drosophila, of course, is unlike Musca domestica, you can start studying it genetically, even though that's, again, when I went in the 80s, was still more of a kind of an article of faith than an actual fact. You can manipulate Drosophila, and therefore it's much more interesting. Plus it doesn't smell as much as Musca, which is kind of vile, lives on rotting meat, so flies just live on porridge and they don't smell quite as... See, flies, there's this kind of reification, the reduction of the whole of Diptera, with however many, 100,000 species, to the fly, which is Drosophila. So there's this, Drosophila melanogaster, as studied in the lab, is not actually an organism. This is the point Köhler makes. It's actually been turned into a piece of laboratory equipment, and what you want from it is as little variation as possible, because you're trying to ask very specific questions about the role of genes on behaviour or whatever character. So the interesting stuff in biology gets ignored, all the variability into intraspecific variability, wild variability. That was a bit of a sidetrack. But it's very important to, and my slip, which I'm calling it the fly, is, I think it's quite indicative of the state of mind of, and how even somebody like me, who is very interested in evolution and is well aware there are plenty of other kinds of flies, I will still slip into that shorthand. To come back to your question about pheromones, so what we were able to do in Drosophila was to isolate very easily, relatively easily, a certain part of the chemical signature that is used in chemical communication. This turns out to be basically used by all insects, probably all arthropods, all insects, I bet will be all terrestrial arthropods, and there's even recently been evidence that it's also used even in aquatic crustaceans, marine crustaceans. So these are compounds that are on the outside of the cuticle, which may have initially been used in terrestrial organisms for prevention of dehydration, because they're like waxes, and then get used by sexual selection to be able to identify which sex is which, and which species is which, and perhaps which fitness, the fitness of a particular individual. Now, these are not like moths. If you think of, most people think about pheromones and chemical communication, you think about very volatile compounds, and moths are able to zero in. A male moth can zero in on a female from a kilometre away. His antennae can detect a single molecule of the female's sex pheromone. And Drosophila stuff isn't like that. It's all about very close contact pheromones. They actually kind of touch each other, and they taste it. It's more like taste than olfaction. But what you can do, and what we were able to do, was to, I've left the field, I don't work in that field now, but what my colleagues have been able to do, and I've continued to publish on, is you can actually create mutants in both the signal and the receptor, because one of the big issues in any form of understanding, any form of communication, is how it can evolve. Because you've got a signal and you've got a receiver, and we know that they differ between species. They may even differ within a species, may have geographical variability. So how can that occur? Because if the signal evolves, it drifts by genetic drift or by some adaptation, the receiver has got to go with it, otherwise the system doesn't work. And so we say, oh well, they're co-adapted, which is just a way of saying, well, they evolved together, but we don't actually, what does that mean? How does that work? And we don't know the answer to that. In moths, for example, in one particular species of moth that's been studied, we know that the signal and the receiver are coded by completely different genes on completely different chromosomes. So it's not even as though these two genes are very tightly linked, so they tend to be inherited together. So how that works is completely obscure. In one example in Drosophila, most interesting example, we know that the one aspect of the signal and one aspect of detection are in fact coded by the same gene. So there are different, they're genetically, developmentally determined expression of this gene, and it's expressed in both the glands that produce the signal, but also in various parts of the brain which are processing that signal. What's odd is that in both cases it's coding for the same thing, which is an enzyme which is involved in the biosynthesis of the compound. So what that's doing in a bit of the brain, we don't know, but it's been very clearly demonstrated that it's exactly the same gene that's responsible for signal and detection. On the other hand, my hunch is that's a very odd example, but at least it provides us with a solution to a major biological problem, which is common to all communication systems, as I say, to how they evolve.